Hello. Very good evening to the student who have joined the class. Can you let me know in the chat box if my voice is audible to you? Also, kindly let me know if the PPT is visible and my video is on. Please let me know in the chat box if everything is alright with the audio, video and the presentation. Okay, thank you so much. We will wait till it is 6 p.m. and more students join, then we will start today's session. Okay, so it is almost 6 p.m. So I think I will start with the introductions. So very good evening to the students who have joined. My name is Mousumi Deb and I am the PMRF fellow from the Department of Chemistry and Chemical Biology, IIT ISM Dhanbad. Today it is the week 12 live session for the NPTEL course of Organic Chemistry in Biology and Drug Development. So we have been doing this kind of live session for past 12 weeks, and also I had one extra class this Sunday on 15th October, for which the video and PPT I have shared with you at 6 p.m. right now. I will wait for 2 more minutes to see if more students are joining, and then I will start the session of today. Okay, so I hope my voice is audible and the video is visible. Okay. Yeah, so okay, I will go with my introductions again. So very good evening to the students who have joined. My name is Mousumi Deb and I am the PMRF fellow from the Department of Chemistry and Chemical Biology, IIT ISM Dhanbad. So I welcome you all in the last live session for the NPTEL course of Organic Chemistry in Biology and Drug Development. So today it is the week 12 live session. It is our last live session. We have been doing this kind of live session for past 11-12 weeks, and we did one extra class on this Sunday, means on 15th October, for which I have been sharing the class recordings and the PPTs with you. I hope you all are aware of that. Like on the NPTEL website, on the SWAM portal, you will find all the lecture videos of the past classes, and the PPT and the lecture material, everything is present there. So you can go through all those things. So today it is our last session for this course. It has been a good journey for the past 12 weeks where we were learning the interfaces of organic chemistry and the biology and the drug development. So today it is our last live session. I hope my voice is audible and the video is clear and as the presentation. So I will just start with the class, and we will wait when more students will join, then we will let them in. Okay. So I think the PPT is clear. So yeah, just I will just summarize these things. So there will be a problem solving session every week, which we have been doing. The classes were from Tuesday for 6 to 8 p.m. and the sessions are live on Google Meet platform. So this thing I want to just clarify that if any network disruption happens, please do not leave the class. I will try to join as soon as possible. The internet connection is little bit slower today, but I will try to my best, and if even my connection is lost, I will try to rejoin as soon as possible. So please wait for some time, do not leave the class. Okay. And if the problem of internet arises from your side, you can rejoin at any time, so there won't be any problem, as I will share this lecture video to you and this PPT also. So during the session you can post your questions and doubt through the chat box. Today it is the last session, so I want all of you to participate, and I want all of you to tell me your doubts. What are your doubts? What are the things you want to me to clarify? Okay, and since today it is the last class, after today's class I won't be able to meet you online, but I will drop my email ID. I think you guys have my email ID. So if you have any doubt, any problem, any query after today also, so I will be available to you, and you can drop an email to me. I will surely answer your query. Okay, since it is the last class, so after this class only you can just connect me through the email. So you can drop an email and I will try to answer your query through the email. And yes, this presentation and class recording will be shared with you soon. So I think I can start today's session. So what we will do in today's session, so today it is a week 12 live session. So we have completed all our 12 classes. In the first class we did for week 1, week 2 all together, and in the last class means on the last Tuesday, what we did is we completed the week 12 course content, right. So today our class will be like a little bit different. So what I will do is, first 1 hour and 1 hour 10 minutes like this, I will give you 12 questions. All these 12 questions are based on your 12 class material, each one question per week, this kind of thing. I have set questions and I want all of you to try to answer these questions, because I want to know if you are revising your last week's things. Okay. So first we will do that 12 question, 12 MCQs, and then I will summarize few topics. So in the Sunday's class, in the last summary class which I had, 1 hour summary class, so in that class what I did was I summarized the things of your course. Like we discussed about the amino acids, we discussed about proteins, a few things about the chromatographical techniques, then we discussed about drug development, we discussed about DNA replication, translation, the enzymes which take part in it, and also we discussed about the pharmacokinetics, pharmacodynamics, and all those topics we discussed in the last day. So today what is remaining I will discuss. So basically today I will discuss about various drugs, okay, so or various biological medicinal compounds regarding. So today I will discuss about your antibacterial agents, antifungal agents, anti-hypertensive agents, antiviral agents, and also I will discuss about the medicines which are used to treat the cholesterol problems and the your antidepressants. Okay, see, antidepressants and this cholesterol medicine, this were not very much included in your course, but these were present, but it was not much mentioned. So I thought to give you an overview, so it will be helpful for you in future also. So that things I will complete after I complete all the 12 questions, then we will start that thing. Okay, so I think I will start. If more students join, then I will let them in. Okay, so the first question of today's session is, which of the following statements is true regarding the movement of the amino acids in gel electrophoresis? Okay, which of the following statements is true regarding the movement of amino acids in gel electrophoresis? Your options are: number A, if pH less than Pi, Pi means your isoelectric point, then it will move toward the negative electrode. Option B is, if pH less than Pi, then it will move toward the positive electrode. Option C, if pH greater than Pi, then it will move toward the negative electrode. Okay, somebody wants to join. Yeah, so I will repeat the first question again. Okay, so the first question is, so today it is a summary class, so I am giving you each question from each week to see if you remember the things we did in the previous sessions, or it will help you to revise the things. Okay, so the first question is, which of the following statements is true regarding the movement of amino acids in gel electrophoresis? Your options are: number A, if pH less than Pi, then it will move toward the negative electrode. Option B, if pH less than Pi, then it will move toward the positive electrode. Option C, if pH greater than Pi, then it will move toward the negative electrode. And option D, if pH equal to Pi, then it will move toward the negative electrode. So which of the following statement is true regarding the movement of amino acid in gel electrophoresis? You have to let me know. Okay. So I am giving you some time, read all the options very carefully, think what is the gel electrophoresis is and how the pH changes with the, how the charge of the amino acid changes with the change of pH. Okay, think all these things carefully and then try to answer this question. I want all of you, whoever is present in the class, all of you to try to answer this question since this is our last class. So I want all of you to participate. Which of the following statements is true regarding the movement of amino acids in gel electrophoresis? Pi is your isoelectric point. Now think what is the Pi is all about and think how the charge of amino acid changes. Whenever you face this kind of questions, it is better to draw a normal structure of an amino acid, okay, and then try to think about how the it will be charged with change of the pH. I will give you some time, think and then write. Okay, let me check the chat box. Okay, so I have three different answers. One is number A, then I have number B and then number A. Means A and B are the most given answers. Okay, let me check which of the following is the correct one and then we will discuss it. So, the correct answer of this question is number A. If pH is less than Pi, then it will move toward the negative electrode. Now, why this is? So, first let us know what is gel electrophoresis. Okay. So, electro means the flow of electricity and phoresis means to carry across. So, electrophoresis is a technique used to separate and sometimes purify macromolecules, especially proteins and nucleic acids that differ in size, charge and conformation. As such, it is one of the most widely used techniques in biochemistry and molecular biology. So, gel electrophoresis is procedure that separates the molecule on the basis of the rate of the movement through the gel under the influence of a electric field. So, in the gel electrophoresis what happens, basically there is a pH gradient, okay, and amino acids will have some specific charges based on their Pi or isoelectric points. Okay, and the amino acids which will be positively charged and at a particular pH it will move toward the negative electrode, and the amino acids which have the negative charge it will move toward the positive electrode. Now how the charge is depending on the your pH. So, the simple rule which you have to remember is, if pH is less than isoelectric point, then that will be present as positively charged species. Okay, and if the pH is greater than isoelectric point, then it will be present as negatively charged species. So, if pH less than Pi, then it means the pH is less than isoelectric point, then it will be present as positively charged species, and if it is a positively charged species, it will move toward the negative electrode. Okay, and if the pH is greater than Pi, okay, it will be present as the negatively charged species, so it will migrate to the positively charged electrode. So, remember this 2 point is very important, it is very important. You may give some, I have given this kind of examples in your previous PPTs. If you read the PPTs, you will find it. So, I have given the examples of the amino acids with the particular PIs, and then The question can be given like this: you are given 4 or 5 amino acids, then you are given the pi and a particular pH is mentioned. You have to identify which amino acids will move toward which electrode. So this kind of question can also be given to you. So this remember, if pH is less than isoelectric point, that will be present as positively charged species, mean it will move toward the negative electrode, and if pH is greater than pi, then it will be present as the negatively charged species, mean it will migrate to the positive electrode. Okay. So this was the first question of today's session. Now moving toward the next problem. So it was from week 1. Okay. So the next question is, which is from week 2. So problem 2 says, just read this problem very carefully and then try to answer. Which of the following is the correct sequence of a hexapeptide based on the following statements? Amino acid composition is 2R ASVY. I won't tell you what is the full form of which amino acids. Okay, just you have to remember the single word term of the single letter code for the amino acids also. So the amino acid composition is 2R, means 2R amino acid, 2A, 1A amino acid, 1S, 1V and 1Y. Sanger reagent, if the Sanger, we treat this particular hexapeptide with Sanger reagent, we will have this fragment DNPA. Then if we do the trypsin digest, we will get 2 peptides, one have RAV, these 3 amino acids, and one have RSY, these 3 amino acids. This is so big thing, one is thing I want to clarify. This is not a particular sequence. Okay, it's not like R will be bonded with A, then it will be bonded with V. It is not a sequence. But if trypsin digest happen, 2 peptide will have RAV, these 3 amino acids. The order may be different, but these 3 amino acids will be present in one peptide, and these 3 amino acids will be present in one peptide. The order is not as same. And chymotrypsin digest will give us 2 peptide. One will have ARVY, this 4 amino acid, and other will have R and S, these 2 amino acid. Now your options are given like this. Option A is like AYVRSR. Option B is AVRYSR. Option C is AVRYRS. And option D is given like AYVRRS. Now just read all these things very carefully. Think about which amino acid and how these things happen, which enzyme cleaves where, and then try to answer this question. I will give you some time for this question. I know it is a little bit tricky, but just read carefully. I think you can do it. Okay. So I want all of you to take time, think about all of this, read very carefully and then answer. Which is the correct statement, correct sequence for this hexapeptide based on the following statements. Read the statements carefully, think and then write. These kind of problems will be given to you, I think, in the examination, because these type of problems are very important to know where the particular enzyme cuts a hexapeptide bond. Read the statements carefully. It says amino acid compositions are 2R, 1A, 1S, 1V and 1Y. The Sanger reagent treatment will give us a fragment DNPA. Trypsin digest will give us 2 peptide. One will have RAV, these 3 amino acid, and other will have RSY, these 3 amino acid. And trimotrypsin digest will give us two peptides, one is ARVY and one has RS. This is not a order of the particular peptide bond peptide chain. Okay. Let me check the chat box to see if there is any answer. Okay, somebody also wants to join. Okay, so okay, I will just give you 2 more minutes for this question. Since we have somebody who joined later. Okay, I will just show you how to do it. Okay, I think this type of question is new to you, so I will show you and how to do it. So first I will repeat the question again. So it says, which of the following is the correct sequence of a hexapeptide based on the following statements? Your options are: amino acid composition is 2R-ASVY. Sanger's reagent, it will give me a fragment which is DNPA. Trypsin digest will give me two peptides which one have RAB, these 3 amino acid, and RSY, these 3 amino acid. And chymotrypsin digest will give me two peptide, one is ARVY and other is RS. So these were your options. So now I will tell you what the answer and how to solve this question. Okay. So let me just see if there is any answer or not once again. So yeah, I think I will tell you how to do it. Okay, I think this is a new kind of question for you, so it's fine if you cannot answer it right now, but let's just see if you are understanding what I am saying or not. So the correct answer will be and number B, AVRYSR. So this is the correct answer. This is the sequence of the hexapeptide. So now let us see how to solve this kind of problem. The first thing is, when you are reading to solve this kind of question, you know about two things. One, what are the one letter abbreviation of amino acid? You have to remember this thing. Which amino acid is which kind of amino acid, like which amino acid is a basic amino acid, which amino acids are acidic amino acid, which have a hydrophobic side chain or which have a aromatic side chain, this kind of things. And the second thing is, you have to remember which enzyme cuts a peptide chain where. Okay, so this is a particularly your application based question. So first we will see the one letter abbreviations of the amino acids. So these are the one letter abbreviation of the amino acids. Okay, so alanine is for A. So the 3 letters are easy. Okay, you know, I know you can just if you see the 3 letter word, you know which one is which one, but one letter abbreviations are little bit sometimes a little bit of difficult to remember few amino acids. Okay, like alanine is A, it is easy. Arginine is R, asparagine is N, aspartic acid is D, cysteine is C, glutamine is Q, glutamic acid is E, glycine is G, histidine is H, isoleucine is I, leucine is L, lysine is K, methionine is M, phenylalanine is F, proline is P, serine is S, threonine is T, tryptophan is W, tyrosine is Y, valine is V, asparagine or aspartic acid is asparagine is B, and glutamine or is Z. Glutamine can be given as Z also and also as a Q. So these are two, just don't think this glutamine and this glutamine, these are some difference this there. So what are the amino acids which are present? So we have 2 arginine, 1 alanine, 1 serine, 1 V is for valine, and 1 Y, and Y stands for your tyrosine. So these are the 4 amino acids which are present. I will highlight it here so it will be easier for you. So we have arginine, we have alanine, we have serine, we have tyrosine, and we have valine, right? So among which we all know arginine is a basic amino acid. Okay, and your tyrosine, it is a amino acid which have a aromatic side chain. So these 2 amino acids are very important to remember. Why? I will just tell you now. So as you can see the composition, so it says the first statement says that the Sanger reagent is giving me DNPA. So what does the Sanger reagent do? So basically in the Sanger methods, Frederick Sanger has demonstrated a method to determine amino acid residue located in the N-terminal end of the peptide chain by using the reagent fluorodinitrobenzene, which is FDNB. Sanger showed that for the first time amino acids are covalently together by alpha amino and alpha carboxyl group. He suggested stepwise release and identification of amino acids starting from N-terminal. He used a reagent fluorodinitrobenzene or FDNB, which is commonly called Sanger's reagent. FDNB reacts with free NH2 group of N-terminus. Upon hydrolysis, a yellow colored dinitrophenol or DNP derivative of N-terminal amino acid is produced. See, Sanger's reagent is giving me N-terminal. So this means, since this is giving me this, so means the N-terminal will have this amino acid A. So A will be present on the N-terminal, right? So as you, if you, now you have to check all your options. So in every option we have A on the N-terminal. So from the first statement we cannot say which is the particular sequence of the hexapeptide, right? So now the second point, we have to come to the second point. Now it say that trypsin digest is giving me two peptide, one is RAV and other is RSY. Now what is trypsin digest? What the trypsin digest do? So basically trypsin breaks the polypeptide chain, okay, to the next to the basic amino acids arginine and lysine. So wherever there is arginine and lysine, it will cleave at the right side, okay. Right side mean the C-terminal side of the particular basic amino acid. So now if you check all your peptide sequence, if we see, if we try to detect which is our correct question, now we have to do the elimination method. So see the first sequence. So where is there is R, so there is R in this middle and this end, okay. So if the trypsin digest happens, so it will break here. So we have two fragments, one is A, Y, VR and another SR. So it is giving me two fragment, but one has 4 amino acid and other has 2 amino acid. So which won't be possible, since our this is wrong, because our clearly our third statement is saying that the trypsin digest will give me two peptide, one has each having 3 amino acids. And if the first in A it breaks on the C-terminal end, so it will give me two fragment, but one have 4 and other have 2. So it is not correct. Option number B, if it breaks here at the right side of the basic amino acid, it won't break here, there is nothing to break here, so it will break only in middle. So now what we will have, we will be having two fragment, right. So we will be having two fragment, one has AVR and other as YSR. If you may see, so this is matching with our statement number 2, right. So it may be our answer, it may be our correct answer. Now the third option, it will break at this place and it will again break at this place. Why? Because we are in middle now, so we have the C-terminal end. So this is giving me 3 fragments trypsin digest. One has 3 amino acid, one has 2 amino acid and one has 1 amino acid only. So it won't also be correct. And now problem number D, if we see, it will also break at the 2 position and it will also give me 3 fragment. One has 4 amino acid, one has 1 amino acid, other has 1. So it won't also be a correct answer. So only correct answer which seems is number B from till now. So now we will just see if it is matching with our last statement. So last statement is says that chymotrypsin digest giving me 2 peptide. One have 4 amino acid ARVY and other has 2 amino acid RS. Now what is a chymotrypsin digest? Basically chymotrypsin breaks the next at the C-terminal of a aromatic amino acid. Aromatic amino acid means phenylalanine, tryptophan, tyrosine. So if you can see here, our sequence is this, okay. So if I just erase this thing here, okay. So this is our sequence. If chymotrypsin digest happens, so what will have, where will it cut? It will cut at the C-terminal end or the right side end of the aromatic amino acid, which is aromatic amino acid is Y. Y is for your tyrosine, okay. So it will break in this position. If it breaks at this position, we are having two peptide, one has 4 amino acid ARVY and other has 2 RS. So this is matching with our last statement. So number B is the correct statement. So now I want all of you, whoever is present, did you understand this problem? Just let me know in the chat box, or I will repeat it. This is a little bit different kind of question, so I want all of you to let me know if you understood this problem or not, or do I need to repeat it? Hello? The students who are present, please let me know if you understood this. Okay. Madhura, if you have any problem, please write in the chat box. I will repeat it once more if you want me. See, just you have, okay. So you have understood it, right? All of you have understood this problem, how I solved it. So if you any problem is given to you like this, I hope you all of you will be able to solve this kind of problem. This is very easy. Okay, so this is very easy, but just you have to see what are the fragments are there, okay, and what are the enzyme digest are mentioned there, which enzymes are cutting where, and just try to eliminate the wrong options first, and then to just check if the whichever what is remaining you think is correct, just check the last one if it is matching with it or not. Okay, if you practice more questions like this, I think you will be able to answer this type of question very easily in your examination. Okay, so just remember this thing, remember one word abbreviation of amino acids, try to remember which enzyme breaks which position, then you will be very easily able to answer this kind of question. So now I will move toward the next problem. Problem number 3 says, which one of the following protein purification process is not based on the charge? Your options are number A, SDS page, number B, ion exchange chromatography, number C, native gel electrophoresis, and number D, isoelectric focusing. So which of the following protein purification process is not based on the charge? SDS page, ion exchange chromatography, native gel electrophoresis, and isoelectric focusing. Everybody think which technique does what, and try to answer this question. I discussed briefly about few techniques of chromatography in the last summary class. I hope you have understand that class and the topics nicely that day. So now think which technique uses which protocol, and then try to answer this question. You have to find out that protein purification process which is not based on the charge. Which one of the following technique is not based on charge? Number A, SDS page, number B, ion exchange chromatography, number C, native gel electrophoresis, number D, isoelectric focusing. Let me check the chat box if there are any answers. Okay, I have few answers here. I am having number B and number C. So I have two answers till now. Okay, let us check which of the following. See, the question was like this. It had said which of the following is not based on the charge. So the correct answer will be SDS page. So SDS page, this technique is based on the molecular weight, not on the charge, the purification. Ion exchange chromatography, I mentioned in the last class that in the ion exchange chromatography what happens, like we are we are having the purification column with the stationary phase is particularly the particular charge is there in the stationary phase, and based on the charge of the proteins, okay, it will get purified. Native gel electrophoresis, electrophoresis is also based on the charge, which we discussed today's class only, right? Because in the gel electrophoresis the species will be charged based on their Pi and pH difference, and then it will move toward the positive negative electrode. Isoelectric focusing is similar to the gel electrophoresis, it will also based on the Pi values and the charge, okay. But SDS page, this is not based on the charge. Now let us just have a little bit of difference. See, so proteins may or may not be may not have a overall charge, okay, but there is no consistent charge to size relationship like we need. So the one protein can be charged or it cannot be charged, okay, and there is no consistent charge to size ratio, this kind of relationship. So we add a particular salt, which is your sodium dodecyl sulfate. Okay, so this have a long hydrophobic chain which is negatively charged and sodium ion as a counter ion. So SDS has a negative charge, it binds with the protein evenly and keeps it linear. So it has a negative charge, so it will bind the protein and keep it linear, okay, and it keeps, so there is a direct charge to length relationship. So now there is a direct charge to the if the protein chain is longer, it will take more SDS and it will be, he wants to join. Okay, so if the particular your protein is a very longer or it has a longer, means it have a more molecular weight, then it will take more SDS and it will have more negative charge. So there is a direct relationship between charge and length right now, okay. So any natural charge it has will get overpowered by the charge coating it gets from SDS. So now we can use this charge to send it to the polyacrylamide gel mesh. Longer things get tangled up to the mesh more, so they travel more slowly, and when you turn off the power you don't move that so far. So basically here what happens, like based on the molecular weight this purification happens, okay. So how based on the molecular weight the purification happens? See, if the molecular weight of protein is high, the chain will be longer, okay, because SDS is making the folded protein linear, so that if the charge is and by adding the negative charge, so here the charge is not the basis of the separation, okay, it only is making the protein chain longer or linear, so that there is a charge to size ratio, okay, which we can use. So if the length is longer, means it has a higher molecular weight, okay, and if it has higher molecular weight, means if the protein is bigger in size, so it will tangled in the polyacrylamide mesh, okay, gel, which is used here, and it will move slowly toward the gel. So the bigger proteins or the bigger, so as you can see here, the bigger ones will be at this side at the negative end and the smaller one will be to the positive end. Okay, so that's how base of, remember SDS page, the purification is not based on the charge, it is only based on the molecular weight of a particular protein. Okay. So now I will move toward the next problem. Problem number 4 says, which inhibition study deals with the decrease in the value of Vmax while Km is unaffected? We all know what is Vmax, what is Km. I discussed it in the last summary class, how the Vmax is calculated and how the Km is calculated in detail. The derivations I did in the last summary class. You can check the PPT of that class if you want to revise. So, which inhibition study deals with the decrease in the value of Vmax while Km is unaffected? Your options are number A, competitive, number B, non-competitive, number C, uncompetitive, and number D, suicide. Which inhibition study deals with the decrease in the value of Vmax while Km is unaffected? Which inhibition study deals with the decrease in the value of Vmax while Km is unaffected? Competitive, non-competitive, uncompetitive, or suicide? Let me check the chat box now. Okay, so I have one answer which is number B. Okay, let me check. See, since this is the last question, I want all of you to participate. Even if your answer is wrong, this doesn't matter, you all know. But I want all of you to participate because the things are from your last class. All these things I have mentioned earlier, so it will help you to revise the things. Okay. So the correct question answer is number B. Very good. Non-competitive will be the correct answer. So now let us see. So all these questions are related with the Lineweaver-Burke plot for inhibition. So what is the Lineweaver-Burke plot? So how this particular plot comes, I have discussed the derivation earlier also and in the last class also. So from the Michaelis-Menten constant, what we have Vmax, Vmax equal to your I just made the mistake here. Just let me erase this thing. So what is your Michaelis-Menten equation? The Michaelis-Menten equation goes like this. So V equal to Vmax, I am writing it as Vm, okay, the Vmax, okay, just let me write the Vmax, concentration of S, the substrate concentration, divided by Km, which is a Michaelis-Menten constant, plus S. This is your Michaelis-Menten equation. And what is the Lineweaver-Burke plot? If you just make the reciprocal of this equation, so we will have this 1 by V, and if you write it as in the elaborate formation, we will have like 1 by, so we will have Km, okay, Km by Vmax, we will have Km by Vmax into 1 by S, and we have plus what it is 1 by, if we reciprocal it, SS will be cancel out and we will have 1 by Vmax, right. So this is the reciprocal, double reciprocal equation or Lineweaver-Burke plot equation, okay. So as if we compare this with y equal to mx plus c, which is the straight line equation, so you can say that y is your y axis, which is your 1 by V, S is 1 by S is the x, okay, and Km by Vmax is your slope, which is mentioned here, and 1 by Km is the intercept value, okay. So now if you check like in which curve, so I have told you to remember, I have mentioned these graphs earlier also. So in the competitive inhibition, the graph look like this, okay. So in this case the Vmax, so the slopes are same, the slope of with inhibitor or without inhibitor is same, only the change is in the the intercept is the same, only the change in the slope, okay. So Km is increased here, so the Vmax is here unaffected, okay, Vmax is unaffected, so the slope is Km by Vmax. So as you can see with inhibitor, the slope is increasing, and Vmax is unaffected, means Km is increasing. So remember, for competitive one, Km is increased, Vmax is unaffected. In the uncompetitive one, the slopes are same, so Km by Vmax ratio is same, but with the inhibitor what happens, with the inhibitor the particular intercept is increasing, okay. Since 1 by Vmax is increasing, means Vmax is decreasing and Km will also reduce, so the ratio is same, means the slope is same. And for the non-competitive one, the Km is unaffected, okay, only the change in the which is in the your Km is unaffected, and only change is the decrease in the Vmax value, because the intercept 1 by Vmax is increasing, means the your Vmax is reduced, and Km will be same in this case because the slopes are different here, okay. So this was your all about your non-competitive, competitive and uncompetitive inhibition, how the graphs look like. So I want all of you to remember these graphs. These graphs are very important. What are the equations for Michaelis-Menten equation, and also what are the equation for the Lineweaver-Burke plot. okay, because mathematical problems will also be given for this equation. Equations won't be mentioned in the question, so you have to remember the equations. The derivations I have mentioned in the last summary class, you can check it from there, okay. If you understand the derivation, you don't need to memorize anything, you can just easily derive the Lineweaver bark plot from the Michaelis-Menten equation easily, because you just have to make the reciprocal of the equation, okay. So this was the problem number 4. I will move toward the next problem. So problem number 5 says, what is the role of the enzyme apirase in the pyrosequencing? Your options are: number A, react with ATP to give light; number B, release ATP from ATP sulfurylase; number C, break down the DNTPase; and number D, make the complementary DNA strands. Pyrosequencing is a technique which I had not I think mentioned in the sessions, but it is in your syllabus, so I thought to give an overview of pyrosequencing. So there is an enzyme called apirase which is very important in this technique of pyrosequencing. You have to identify which is the particular role of the enzyme. Options are: number A, react with the ATP to give light; number B, release ATP from ATP sulfurylase; number C, break down the DNTPase; and number D, make the complementary DNA strands. What is the role of the enzyme apirase in pyrosequencing? Okay, let me check if there is any answer, or I will discuss this thing. I have one answer which says number D. Okay, let me check if it is correct or not, and let us discuss. So the role of the enzyme apirase in pyrosequencing is number C, break down the DNTPase. Now let us see what is pyrosequencing. So pyrosequencing is a method of DNA sequencing based on the sequencing by synthesis principle. So we have discussed like the sequencing of your Sanger sequencing, and we also discussed about Maxim Gilbert sequencing, which I think I have mentioned. So pyrosequencing is a similar technique. It differs from Sanger sequencing, relying on the detection of the pyrophosphate relase, since the name the phosphate on nucleotide incorporation rather than the chain termination with dideoxynucleotide. Single strand DNA template is hybridized to a sequencing primer, incubated with the enzyme DNA polymerase, ATP sulfurylase, luciferase, apirase, and with the substrate adenosine 5-pyrosulfate, APS and luciferin. So how this thing happens? So step 1, the step 1 is the DNA segment is amplified, one strand is biotinylated strand is hybridized to sequencing primer. Primer plus template is incubated with DNA polymerase, ATP sulfurylase, luciferase and apirase. Add DNTPase, if complementary to base, DNA polymerase will add the DNTP to sequencing primer. For each DNTP incorporated, an equimolar pyrophosphate will be added. PPI, now step 2 is, so we have APS and PPI, the from ATP with the sulfurylase. So PPI will be converted to ATP. In the presence of ATP, luciferin will be converted to oxyluciferin. This reaction generates light. The light is detected by the sensor, peak is created. The height of the peak is proportional to the amount of DNTP incorporated. Okay, so we have this primer plus template which is incubated with DNA polymerase, ATP sulfurase, luciferase and apirase. So all the enzymes. So the height of the peak will be proportional to the amount of DNTP. Now apirase enzyme, in the step 3, what will happen? Apirase degrades the unincorporated DNTPase and ATP and go back to step 1. So this is a cyclic process happens. As the process continue, complementary DNA sequence is elongated, nucleotide sequence is determined from the peak of the pyrogram. So this is the overview of pyro sequencing. I don't think you have to go to the details of the method. Just remember what are the enzymes which are involved in pyro sequencing, like DNA polymerase, ATP sulfurylase, luciferase, apirase, and what are the roles of the each and every enzyme. Remembering this much I think will be enough for your course syllabus. Okay, and Maxam Gilbert and Sanger sequencing methods I didn't included in today's class, but these two topics are also very important. I want all of you to just go through the previous slides and read that particular sequencing methods also. Okay, so now I will move toward the next problem. Problem number 6 says, what would be the complementary RNA strain for the following coding DNA sequence in the transcription process? The DNA sequence is 5 prime GCG TA TG 3 prime. Your options are number A 5 prime GCG UA UG 3 prime, number B 3 prime CGCA UAC 5 prime, number C 5 prime CGCA UAC 3 prime, and number D 3 prime GCGUA UG 5 prime. This DNA strand, coding strand and non-coding strand, these question I have given you earlier. Okay, so I want all of you present in the class to answer this question, because this is very important thing and all of you get confused what is the coding strand, what is the non-coding strand, what is the template strand, what is the non-template strand. So there is always a confusion regarding this, and I think I have somebody has mailed me regarding this problem also, that they were confused about what is the coding and what is the non-coding strand. So that's why I thought to give this question, because it will help you to remember what are the strand is. Now try to answer the question. I want all of you to participate, read the strands carefully and then identify which of the following is the correct one. What would be the complementary RNA strand for the following coding DNA sequence in the transcription process? Let me check the chat box. Okay, so I have two answers. I have one answer, number D. I wanted all of you to answer this question. I have mentioned this earlier also. Okay, let me discuss. See, that's why these things you need to revise very well. So the correct answer will be number A. Okay, 5 prime GCGU AUG 3 prime. So if you see, just look at this carefully. One DNA strand is from 5 prime to 3 prime. The strand which is from 5 prime to 3 prime, it is the non-template or the coding strand or the sense strand. And the strand which is from 3 prime to 5 prime, it is the template strand or non-coding strand or antisense strand. So, based on the template strand or the non-coding or antisense strand, the RNA primer, the RNA complementary RNA strand will be synthesized from 5 prime to 3 prime. So, this RNA strand will have a complementary base pairs with this non-coding strand. So, one thing you can do is convert the coding to non-coding by writing the complementary strand. So, what will be the, if this is the coding strand, what will be the non-coding strand? The non-coding strand will be 3 prime. Okay. Then we will have C, G, C, A, T, A, G, sorry not G, it will be C, just let me erase, A, C, 5 prime. So, this is the first you have to convert coding to non-coding, then from coding to from this non-coding you have to write the complementary strand for the RNA. So, what will be the complementary one in the RNA? It will be 5 prime, G, C, G, A. For A, what will be the complementary? It will be uracil, right? Then for T it will be A, then from A it will be U, then G, then 3 prime. So, remember we have, if you are given the coding strand, you have to convert it to the non-coding and then write the particular RNA strand. If you are given non-coding, just you have to directly give the complementary ones. Okay. So, remember which strand is which strands. Non-template coding sense strand is the 5 prime to 3 prime, and template non-coding or antisense strand is the your 3 prime to 5 prime. So, from DNA, mRNA is synthesized by transcription, and from mRNA the protein is synthesized by the method of translation. So, this is the sense strand which is 5 prime to 3 prime, antisense strand from 3 prime to 5 prime. From the antisense strand we have this RNA, mRNA with from the 5 prime to 3 prime with codons, and with the codons we will have the tRNA with the anticodons. Okay, and from this anticodon we have this peptide. Okay, so this is the whole thing. So, it is very important to remember which is sense strand, which is antisense strand, and what is mRNA codon, which is tRNA codon. Okay, so don't get confused, read it for couple of times, practice this kind of problems couple of times. So, I think you will be able to answer this with ease. So, now next problem. Problem number 7 says the palindrome sequence recognized by restriction enzyme E. coli is number A, AAGCTT, number B, CTGCAG, number C, GATATC, and number D, GAATTC. So, the palindrome sequence which is recognized by restriction enzyme E. coli is, identify this in the chat box. Which palindrome sequence is recognized by restriction enzyme E. coli? We discussed about the restriction enzymes, what they do and the types earlier also. Restriction enzymes are also a very important topic of your course. Which palindrome sequence is recognized by restriction enzyme E. coli? Let me check the chat box. Okay. Okay, yeah, I have one answer, number B. Okay, let me check if number B is correct or not. So, the palindrome syndrome which is recognized by E. coli is number D, GAATTC. So, this is the table which shows where the different restriction endonuclease or restriction enzymes cleave DNA. The abbreviation R represents the purine and pyrimidines are represented Y. W represents thymine or adenine. So, E. coli, as you can see, it cleaves between the A and G. Okay, so you have the palindrome syndrome which is recognized by it is 5 prime GAATTC 3 prime or 3 prime CTTAAG. Okay, so basically remember that the E. coli, it cleaves between G and A. Eco RV, it cleaves between the A and T. Okay, so here it will cleave at T and A. This eco RV, it is cleaves this is the palindrome syndrome which is recognized by this. So, and this all these enzymes particularly breaks at each side, like hint 3, it basically cleaves between A and A. Okay, and this HAE3, it cleaves between G and C. So, here each and every enzyme cleaves at a particular site. So, just try to remember what are the sites and also try to remember what are the palindrome syndromes which are recognized by a particular enzyme. So, problem number 8 says which of the following statements is false regarding methotrexate. Your options are: number A, it is a potent anti-cancer drug. Number B, it works by catalyzing the dehydrofolate reductase. Number C, the interaction is non-covalent. Number D, its crystal structure is known. So, which one of the following statement is false regarding the methotrexate? Number A, it is a potent anti-cancer drug. Number B, it works by catalyzing the dehydrofolate reductase. Number C, the interaction is non-covalent. Number D, its crystal structure is known. Which is the statement is false regarding this drug methotrexate? Try to answer this question in the chat box. Read options carefully, think and then write. All of you try to answer the question in the chat box. We have read about methotrexate, just try to remember. Let me check the chat box. Okay, number C. I think I have one answer which says it is number C. Okay, let me check. So, the correct answer will be number B. It works by catalyzing dehydrofolate reductase. This statement is false regarding the methotrexate. So, this is the structure of the methotrexate. Okay, its interaction is non-covalent, it is a potent anti-cancer drug and the crystal structure is also known. The false statement which says it catalyzes the particular enzyme dehydrofolate reductase, actually this drug, it inhibits this this particular enzyme dehydrofolate reductase. So, what is the role of dehydrofolate reductase? We have discussed it earlier also. So, this is the folic acid metabolism. The para-aminobenzoic acid and pteridine, it gives us the dehydrofolic acid, dehydroteroic acid, and the enzyme here is the dehydropteroid synthesis. Okay, and then dihydroteroic acid goes to the dihydrofolic acid by the dihydrofolate synthetase, and dihydrofolic acid goes to the tetrahydrofolic acid, which is the active form of folic acid by the dehydrofolate reductase enzyme. Trimethoprim, methotrexate, these drugs, what they do, they inhibit this particular last step of folic acid metabolism, which is the conversion from dehydrofolic acid to tetrahydrofolic acid by the means of inhibition of the dehydrofolate reductase. Okay. So, the statement number B, which says it works by catalyzing the dehydrofolate reductase, it is false. It inhibits the dehydrofolate reductase. So, MTX or methotrexate is an antifolate belonging to anti-metabolite class of anti-neoplastic agents. MTX is a cell cycle specific chemotherapeutic agents that acts on the S phase and thus inhibit the DNA synthesis. So, this is the conversion. Okay. This is your this is folic acid metabolism. So, this methotrexate, it inhibits this particular thing. THF included in the 2 stages of biosynthesis of purine, and this tetrahydrofolic acid included in the 2 step in the biosynthesis of purine and 1 stage in the synthesis of pyrimidine. Okay. So, this was the problem number 8. So, now problem number 9, it says that in which of the following process the protein 3D structure of the target is must requirement. Your options are number A, ligand based drug discovery, option B, structure based drug discovery, option C, fragment based drug discovery, and option D, all of the above. So, in which of the following process the protein 3D structure of the target is a must requirement? Try to answer the question in the chat box. All of you please participate, even if your answer is wrong, it does not matter, but all of you, I think it is our last class, so I want all of you to participate. In which of the following process the protein 3D structure of the target is a must requirement? Number A, ligand based drug discovery, LBDD. Number B, structure based drug discovery, SBDD. Number C, fragment based drug discovery, FBDD. And number D, all of the above. In which of the following process protein 3D structure of the target is a must requirement? Ligand based drug discovery, structure based drug discovery, fragment based drug discovery, or all of the above? Somebody wants to join, and also I have to check the answers. Okay, so good. Number B is given answer by all of you. Now let me check. So the correct answer will be, very good, number B, structure based drug discovery. So now there are various kind of drug discovery. One is structure based, where the receptor 3D structure is known, generation of the interaction size for the ligand. There is ligand based drug discovery, which have the receptor 3D structure is unknown, and generation of pseudo receptor or direct similarity search. And both of they we also have atom based and fragment based drug discovery. For atom based drug discovery, we need a seed atom and higher number of generated structure with the questionable chemical accessibility. And the fragment based drug discovery, a seed fragment is required, lower the number of generated structures with the good chemical accessibility. Okay. So, what is the workflow of the SBDD project? So, first the crystal structure of the protein available from the literature, it will go throughout the high throughput virtual screening. Then identify the heats will be identified, synthesized or purchase the heats, assay the heats, identify active compound and lead optimization. So, it is a basic workflow of a SBDD project. So, remember the protein's 3D structure for the target is must requirement for a structure based drug discovery. Okay. So, now I will move toward the next problem. Problem number 10 says, which of the following resistant mechanism describes the function of beta lactamase? Your options are number A, efflux pump, number B, target mimicry, number C, drug inactivation and number D, target overproduction. So, what is the following resistant mechanism that describes the function of beta lactamase? Try to identify the correct statement in the chat box. All of you try to remember and try to answer which of the following resistant mechanisms describes the function of beta lactamase. Your options are efflux pump, target mimicry, drug inactivation, target overproduction. Which of the following resistant mechanism describes the function of beta lactamase? Let me check the chat box now. I have few answers. Number C. So, number C is the most given answer. Let me check if the number C is correct or not. So, the correct answer will be, very good, number C, drug inactivation. So, there is a type of resistance called drug inactivation. Some bacteria can use different method to render the antibiotic useless, essentially by destroying the antibiotic structure. So, as you can see in the case of beta lactamase, it inactivates the drug. Okay. So, as you can see in the beta lactam ring is present in your antibiotics, the beta lactam antibiotic like your penicillin, cephalosporin, all these have this beta lactam ring. And this beta lactamase enzyme, what does it do? It hydrolyzes this particularly beta lactam ring, which is very important for the drug effect of the antibiotic effect. Okay. So, this is the mechanism of the drug inactivation. So, there are different kind of beta lactamase. One is the serine beta lactamase, which have this particular serine group, which acts as a member, which the group hydroxyl group of serine, it attacks on this carbonyl carbon of beta lactam ring, and this cleaves the particular ring, and by making this hydrolyzing this beta lactam ring and making this inactivate. And we have metallobeta lactamase, where we have metal bound OH group, which is acts as a nucleophile, attacks the carbonyl carbon and cleaves the ring. Okay. So, drug inactivation is the correct term. So, now problem number 11 say, acyclovir, the structure is shown below, is an inhibitor of DNA polymerase selectively in the virus infected cells. This is because number A, acyclovir is first converted to monophosphate only by the viral thymidine kinase. Number B, acyclovir does not enter the infected cell. Number C, acyclovir first converted to monophosphate by only host thymidine kinase. And number D, acyclovir triphosphate is not accepted by host DNA polymerase. So, this particular drug is an inhibitor of DNA polymerase of the virus. Okay, only viruses DNA polymerase is affected by this drug. What is the reason behind this? You have to identify. Read all the option carefully and let me know in the chat box. All of you try think which of the following can be the correct answer. Number A, acyclovir first converted to monophosphate only by the viral thymidine kinase. Number B, acyclovir does not enter the infected cells. Number C, acyclovir is first converted to monophosphate only by the host thymidine kinase. And number D, acyclovir triphosphate is not accepted by host DNA polymerase. Try to answer the question in the chat box. All of you try to answer the question in the chat box. Okay, let me check the chat box now. Okay, so I have few answers. Number C is the given answer. Let me check. The correct answer is number A. Acyclovir is first converted to monophosphate only by viral thymidine kinase. Okay, so this acyclovir, it is actually a prodrug. Prodrug means it is not a the active form of the drug, but the active form of the drug is the your acyclovir monophosphate. Okay, so first what happens, this acyclovir, it is administered, it is converted to acyclovir monophosphate by the your herpes specific thymidine kinase or viral thymidine kinase, and then the cellular kinase will convert it into the acyclovir triphosphate, and this acyclovir triphosphate will get incorporated in the viral DNA, stops the lengthening the DNA strand, and the terminated DNA inhibits the DNA polymerase in irreversibility, and also it inhibits the herpes virus DNA polymerase competitively. Okay. So, just first statement was the correct one. It is first converted to monophosphate only by viral thymidine kinase, and that is why it is a specific inhibitor of DNA polymerase of the virus infected cell, not the normal healthy cells. Okay. So, now the last question of today's session. Problem number 12 says which enzyme is involved in the activation of the prodrug of cyclophosphoramide. Your options are number A, cytochrome P450, number B, MMO, number C, cytochrome C oxidase, and number D, SOD. So, which of the enzyme is involved in the activation of product cyclophosphoramide? We have discussed about cyclophosphoramide in the last week itself. We have discussed about cyclophosphoramide in the last class, means the last week class. So, which enzyme is involved in the activation of prodrug cyclophosphoramide? Your options are cytochrome P450, number B, MMO, number C, cytochrome C oxidase, and number D, SOD. This is the last question of today's session. After that, I will summarize the topics which we left in the last summary class, which we did not complete it. Now we will complete that topics. So, but before that, this is the last question of today's session. So, all of you try to answer this question. All of you answer this question. Which enzyme is involved in the activation of prodrug cyclophosphamide? Cytochrome P450, MMO, cytochrome C oxidase or SOD. Let me check the chat box now. Okay, so I think all of you have written it is number A. Let me check if number A is the correct one or not. So, the correct answer of the last question will be very good. Number A, cytochrome P450. So, mechanism of action of cyclophosphamide is most commonly used alkylating agent. Both cyclophosphamide and ifosfamide are first biotransformed to hydroxylated intermediates primary in the liver by the cytochrome P450 system. Hydroxylated intermediates then undergo breakdown to form the active compounds phosphoramide mustard and acrolein. Reaction of the phosphoramide mustard with DNA is considered to be cytotoxic step. So, this is the cyclophosphoramide. First cytochrome P450 enzymes will convert it to the active form, okay, and this involves in the first it will be hydroxylated and then the ring opening will give me the aldehyde of alcohol. The non-enzymatic transformation will give us phosphoramide mustard, which is the active agent, and then it will give the reactive species and the acrolein, which is the toxic. Both this reactive species and acrolein is toxic for the DNA. So, that is how this prodrug of cyclophosphamide gets activated by cytochrome P450. So, now we have finished 12 question. All the 12 question will be was from your 12-week course content. Okay, so now what I will do is I will summarize few topics. So, in last class what we did is summarize, we did summarize the amino acids, the proteins, we summarized the chromatographic technique, DNA replication, and we summarized the protein translation, what are the roles of the enzymes. Also we go through with the enzyme kinetics, we discussed about drug discovery, what is the drug discovery, and what are the assays, we discussed about assays, pharmacokinetic and pharmacodynamics basically. So, today I will summarize the topics of which what are your drug agents. So, we will discuss about this. So, this will only I will discuss so that you remember the topics we discussed earlier. So, first thing which we will discuss is your antibiotics. So, antibiotics there are several classes of antibiotics, right. So, remembering each and every class of antibiotic, what are the mechanism of action of the particular class and example, these are the very important thing which you have to do. Okay. So, we have beta-lactam antibiotics like penicillins and cephalosporins which have this beta-lactam ring. Okay. And the examples of penicillin is penicillin G, amoxicillin, triclosan and piperacillin. Cephalosporins, the example is cephalotin, cefuroxime, and all these things. You do not need to remember each and every name, but few names if you remember it is good for you, okay, because the question can be directly given as the drug name and you may need to identify which is the correct class of antibiotic of the drug. Then we have quinolones. Quinolones have we have nalidixic acid, ciprofloxacin, levofloxacin, moxifloxacin. Okay. Then we have macrolide like erythromycin, clarithromycin, telithromycin, and we have tetracycline like oxytetracycline, doxycycline, tigracycline, right. So, the question can be given like nalidixic acid is which class of antibiotic. So, you have to write the quinolone. So, as you can say this is the structure of quinolone. Now, remembering the basic structure is also important because a particular overall whole this structure can be given and you can be asked to write the particular class of the antibiotic. So, just remember if there is a beta-lactam ring present, this is the beta-lactam antibiotics. If the ring is present, these are quinolones. If a particular macrolide cycle, the macrocycle is present, then it is a macrolide. Then if four rings are joined together, it will be tetracycline. So, that is how you have to remember. So, now the most important thing is remembering the mechanism of action of the particular drug. So, this is a summary of the mechanism of action of antibiotics. So first is the cell wall synthesis. So the antibiotics which inhibit the cell wall synthesis of the bacteria are the cycloserine, vancomycin, bactricillin, penicillins, cephalosporins, monobactam, carbapenems. Okay, so these agents, they inhibit the cell wall biosynthesis of the bacteria. This is a bacterial cell. Okay. Then folic acid metabolism, inhibition of the folic acid metabolism is done by your trimethoprim and sulfonamides. Okay. So from the para-aminobenzoic acid to DHF, which is dihydrofolic acid, this conversion is inhibited by the sulfonamides, and from the dihydrofolic acid to tetrahydrofolic acid, this conversion is inhibited by your trimethoprim and methotrexate. Okay, so these are inhibitors of the folic acid metabolism. Then cytoplasmic membrane structure and function, the agents which disrupt this thing are the polymycins and daptomycins. Okay, then we have this cytoplasmic membrane. This is the cell, this is the cytoplasmic membrane, this bluish one. The cell wall is the outer coat, and this is the your lipid biosynthesis inhibited by the plantainmycin. Okay, and then we have protein synthesis inhibition, the inhibition of tRNA like mupirocin and puromycin. Then we have protein synthesis inhibitor. So first we have this 30S inhibitor. So we all know the bacterial ribosome or prokaryotic ribosome have two subunit, one is 50S subunit and one is 30S subunit. So the inhibition of the 30S subunit, all 30S inhibitors are like tetracyclines, spectiomycin, streptomycin, gentamicin, kanamycin, amikacin and nitrofurans. Okay, and the inhibitors for the protein synthesis of 50S inhibitors are erythromycin or like macrolides, chloramphenicol, clindamycin and linacomycin. Okay, and then we have DNA directed RNA polymerase inhibitors like rifamycin and streptovaricins. We have RNA elongation, the agents that inhibit this, which is actinomycin, and we have inhibitors which actually acts on DNA guidase, which are the nalidixic acid, ciprofloxacin, basically which are quinolones and novobiosins. So these are the basically the particular mechanism of action by which the antibiotics works through, and so you have to remember the mechanism of action of the several classes and the examples. Okay. So this was about your antibiotics. So now antifungal drugs. So antifungal drugs was not directly present in your course, but I thought if you are reading so many drug agents, so why not antifungal agents also. So antifungal drugs are medicines deployed in the treatment of mycosis. Fungal infection of the skin are, as of 2010, the fourth most common disease in the world, affecting up to 1 billion people. So there are different kinds of classes like imidazole containing antifungal agents like clotrimazole, oxyconazole, miconazole, iconazole, ticonazole, ketoconazole. So these antifungal agents have imidazole chain, imidazole ring. Okay. So triazole containing antifungal agents are fluconazole, itraconazole, tarconazole, voriconazole, isavuconazole and posaconazole. Then we have griseofulvin inhibits the mitosis in the dermatophytes. It is the ineffective when applied topically. Now we have flucytosine, pyrimidine analog converted to 5-fluorouracil by the fungal enzyme cytosine deaminase, active against the yeast infection. We have polyenes which have natamycin, then amphotericin, neastin. Allylamines like amorphine, natamorphine, terbinafine. We have echinocandins, which is caspofungin, anidulafungin and micofungin. This inhibit the cell wall synthesis by targeting glucans. Okay, and allylamines, they inhibit the squalene epoxide. And azole antifungals, the imidazole and triazole containing antifungals, they inhibit the lanosterol 14 alpha demethylase, the enzyme required to convert lanosterol to ergosterol. So this, remembering this is important, the mechanism of action of the azole antifungals, because most of the antifungals contain the azole functional groups or azole rings as their active component. So mycosis is a fungal infection of the animals including human. Mycosis are common in variety of environmental and physiochemical conditions that can contribute to the development of fungal diseases. Triazoles are newer less toxic and more effective antifungal drugs like fluconazole, itraconazole, albuconazole, posaconazole and voriconazole. So, this is the triazole, this is the 1, 2, 3 triazole. Okay, as this is 1 to 4 triazole rings are present here. So, fluconazole, this is the voriconazole and this is the albuconazole. Like other imidazole and triazole class antifungals, fluconazole inhibits the fungal cytochrome P450 enzyme 14 alpha demethylase. Mammalian demethylase activity is much less sensitive to fluconazole than fungal demethylase. This inhibition prevents the conversion of lanosterol to ergosterol, an essential component of the fungal cytoplasmic membrane, and subsequent accumulation of 14 alpha methylsterols. Fluconazole is an inhibitor of the human cytochrome P450 system, particularly in the isozyme cytochrome P2C19 or cytochrome P3A4 and cytochrome P2C9 to lesser extent. In theory, therefore, fluconazole decreases the metabolism, increases the concentration of any drug metabolized by these enzymes. In addition, its potential effect on QT interval increases the risk of cardiac arrhythmia if used concurrently with the other drugs that prolong the QT interval. Okay. So, now I will move forward the next one. So, hypertensive agent. So, there are different kind of antihypertensive agents which I will tell you with the examples also. So, remember, so what you have to remember is you have to remember the mechanism of actions with few examples. Okay. For every class of the drugs, mechanism of action of the classification, mechanism of action of each class and example of few of the each class is important. So, antihypertensive agents include the diuretics. So, diuretics are also called the water pills. These are the medication designed to increase the amount of water and salt expelled from the body as urine. They are often prescribed to help the treat blood pressure. For example, furosemide, this sold under the brand name of Lasix among others, is a loop diuretic. This medication used to treat fluid buildup due to the heart failure, liver scarring or kidney disease. It may also be used for the treatment of for high blood pressure. Furosemide was patented in 1959 and a health organization of essential medicine approved for the medical use in the 1964. It is on the okay. So, then there is a amidolide. Amidolide is a very important diuretic. Okay. So, this is sold under the trade name of Midamor among the others. It is a medication typically used with the other medications to treat high blood pressure or swelling due to the heart failure and cirrhosis of the liver. So, remember diuretics, it is called water pills, are medication designed to increase the amount of water and salt expelled from the body. Since the volume of the blood is depend on the amount of water and salt. So, lesser the it will lessen the volume amount of water and salt in our body. So, it will be lessen the blood volume. So, it will lessen the blood pressure. Okay. Then we have ACE inhibitor. ACE inhibitors are a very important class of antihypertensive drugs. Angiotensin converting enzyme inhibitor or ACE inhibitor are a class of medication used primarily for the treatment of high blood pressure and heart failure. They work by causing relaxation of the blood vessels as well as decrease the blood volume, which leads to the lower the blood pressure and decrease oxygen demand from the heart. ACE inhibitors inhibit the activity of angiotensin converting enzyme, an important component of the renin-angiotensin system liable to convert angiotensin 1 to angiotensin 2 and hydrolase bradykinin. Therefore, ACE inhibitors decrease the formation of the angiotensin 2, a vasoconstrictor, and increase the level of the bradykinin, a peptide vasodilator. This combination is synergistic in lowering blood pressure. Their example of ACE inhibitor drug is captopril. This is sold under the trade name of Capoten, and it is an angiotensin converting enzyme inhibitor used for treatment of hypertension and some types of congestive heart failure. Then we have AT1 blockers. So, angiotensin 2 receptor blockers, ARBs, formerly angiotensin 2 receptor 1 AT1 antagonist, are known as angiotensin receptor blocker, angiotensin 2 receptor antagonist or AT1 receptor antagonist. These are the group of the pharmaceutical that bind to and inhibit the angiotensin 2 receptor 1 or AT1 and thereby block the arteriolar contraction and sodium retention effect of the renin-angiotensin system. Their main uses are the treatment of hypertension, high blood pressure, diabetic neuropathy, kidney damage due to diabetes, and congestive heart failure. They selectively block the activation of the AT1 receptor, preventing the binding of angiotensin 2 compared to ACE inhibitors. The substances are AT1 receptor antagonists, that is, they block the activation of the angiotensin 2 AT1 receptors. AT1 receptors are found in smooth muscles of the vessels, cortical cells of the adrenal gland and the adrenergic nerve synapses. Blockage of the AT1 receptors directly causes vasodilation, reduces secretion of vasopressin, and reduces production and secretion of aldosterone among the actions. The combined effect reduces the blood pressure. Losartan, irbesartan, omnesartan, so these are the drugs which include the tetrazole group, are the example of AT1 blocker. And then we have the beta blockers like atenolol. This is a beta blocker that affects the heart and circulation, blood flow through the arteries and veins. Atenolol is used to treat angina, chest pain, hypertension, that is high blood pressure, is used to lower the risk of death after a heart attack. Nebivolol is a novel, highly cardioselective beta blocker with antihypertensive efficacy similar to that of the other beta blocker, but with a tolerability better than older agents in its class, which may permit nebivolol to be used more widely and effectively than other beta blocker. Metoprolol is a beta blocker that affects the heart and circulation, blood flow through the arteries and veins. Metoprolol is used to treat angina, which is chest pain and hypertension, and this also is used to lower your risk of death or needing the hospitalized for the heart failure. So this was the 4 very important class of antihypertensive drug. Just read the mechanism of actions, and I will suggest that ACE inhibitors are very important for your course, also the diuretics and beta blockers. So remember these things, it will be enough for you. Okay. Then antiviral drugs. So HIV-1, we will discuss HIV agents here most importantly. So HIV-1 is the most common type of human immunodeficiency virus. It attacks your body's immune system. The virus destroys the CD4 cells. These cells help your body fight infections. Okay, HIV-1 can severely damage your immune system and lead to acquired immune deficiency syndrome, which is AIDS. CD4 cells are white blood cells that play an important role in the immune system. Your CD4 cell count gives you an indication of the health of your immune system, your body's natural defense system against the pathogen, infection and illness. Currently, there is no cure for the HIV or that is AIDS. Once you have the infection, your body can get rid of it. However, there are many medications that can control HIV and prevent complication. These medications are called the antiretroviral therapy or ART. Okay, so HIV is a very one of a very deadly viral diseases, we all know, and because it affects the body's immune system, and we all know CD4 cells are white blood cell, and if you remember the CD4 receptors which this viruses are used for binding to a particular healthy human cell. Okay, so since we are discussing the viral drugs, so I want to make you remember what are the difference between the bacteria and virus. What are the viruses cannot replicate on its own, or it is not a living agent. Okay, it needs the host biological systems to get activated, or for survival it needs the host cell. Out of the host cell, they do not have any particular role, okay, or the existence, but the bacteria can live of its own. We have bacteria in the air also, in the soil also, everything. So bacteria are considered as a living organism. So this is the differences between the viruses and also bacteria. These things I want all of you to remember. So there are different kind of antiviral agents, basically drugs for the HIV. So first we have the reverse transcriptase inhibitors. So reverse transcriptase or RT is an RNA dependent DNA polymerase that utilizes a strand of RNA to synthesize double stranded viral DNA. This mechanism helps the retroviral gene to get incorporated into the host cell's gene. Various RT inhibitors are developed which on the basis of the binding site and the chemical class of compounds are classified into NRTI, nucleoside reverse transcriptase inhibitor, and NNRTI, non-nucleoside reverse transcriptase inhibitor. We will discuss these two classes separately. So, there are also different kind of class of drug. So, protease inhibitors, protease are the enzyme that cleave protein molecule into smaller fragment. HIV protease is vital for both viral replication within the cell and release of the mature viral particle from the infected cell. Various protease inhibitors are developed for HIV treatment. For some FDA approved drugs of the class are saquanavir, indinavir, atazanavir, ritonavir, nelfinavir, amprevnavir and lopinavir, tipranavir. So, these are the example for the protease inhibitor. Now, you can think of it like your human, since these drugs are designed for the particular your viral enzyme. So, why they do not attack the human enzymes also? Because we have this protease enzyme in our normal healthy living cells also. We have this reverse transcriptase also in our healthy cells. So, what will happen? So, how to do it? How the drugs do it, right? So, this is a common question for anti-cancer drugs also, like there is a healthy cell or there is a cancer cell. So, why the drugs only attack the cancer cell, not the healthy cell? So, the reason is basically this: whenever this kind of drugs are designed and synthesized, so there are several differences between the healthy cell and the virus infected cell or the virus enzymes. Okay, like the viral protease enzyme and human protease enzyme, there are several differences. So, the drugs are designed in a way that it will target only, it will take advantage of those differences and it will target only the infected cells or only the virus enzymes, not the normal healthy human cells or the host enzymes. Okay. So, that is why they selectively cleave or they selectively acts on the particular infected cells. So, reverse transcriptase inhibitors are of two class. I have told you, one is non-nucleoside reverse transcriptase inhibitor and which is your NNRTI. Okay. So, among HIV-1 RT inhibitors, non-nucleoside reverse transcriptase inhibitors or NNRTI have gained a definitive place due to their unique antiviral potency, high specificity, low toxicity in antiretroviral combination therapy used to treat HIV. Okay. And then we have NRTIs, which are the nucleoside reverse transcription inhibitors. While NRTIs and NNRTIs alike are effective in terminating DNA synthesis and HIV replication, nucleoside reverse transcription inhibitor NRTIs inhibit reverse transcription by causing chain termination after they have been incorporated into the viral DNA. This was first group antiretroviral agent to be used against HIV. NRTIs lack the 3-hydroxyl group on the deoxyribose moiety. As a result, following incorporation of an NRTI, the next incoming deoxynucleotide cannot form the next 5' to 3' phosphodiester bond needed to extend DNA chain. Thus, when a NRTI is incorporated, viral DNA synthesis is halted, a process known as chain termination. All NRTIs are classified as a competitive substrate inhibitor. So, this statement is very important. We have read about the competitive inhibitor non-competitive. So, this question can be given like this: NRTIs are which class of inhibitors? The options can be given like, is it competitive, non-competitive, uncompetitive? Okay, suicide. This kind of options can be given. So, remember, all NRTIs are classified as competitive enzyme, competitive substrate inhibitors. So, these were the classification for your antiviral drugs, especially the drugs which target the HIV. So, cholesterol. So, now I will discuss the drugs which are lowers the cholesterol levels. So, cholesterol is a waxy fat-like substance that is found in all the cells of your body. Your body needs some cholesterol to make hormones, vitamin D and substances that help you to digest foods. Your body makes all cholesterol it needs. LDL, okay, which is the low density lipoprotein and is considered as the bad cholesterol because it contributes to the fatty acid builds up in the arteries, and which is known as the arteriosclerosis. Okay, so this is the structure of the cholesterol. As you can see, there is a this is kind of steroid. Okay, there is a 4 ring, 6 member, 1 3 member, and this kind of structure, and we have so many chiral centers in the cholesterols. So, two type of cholesterol are there, low density lipoprotein cholesterol and high density lipoprotein, which are HDL cholesterol, is known as good cholesterol because it helps to remove other forms of cholesterol from your bloodstream. Higher level of HDL cholesterol are associated with lower of the heart disease. So, whenever you are eating some food or buying a food substance, if you look at carefully at the back side, there is mentioned what is the amount of LDL, what is the amount of HDL. So, remember LDL is the bad cholesterol. You have to low amount of this cholesterol because it contributes to fatty buildup in the arteries, and high density cholesterol HDL is the good cholesterol, which helps to lower the risk of the heart disease. Okay. Like LDL, VLDL, very low density lipoprotein cholesterol, is also bad cholesterol. Triglycerides are the type of the fat or the lipid found in your blood. When you eat, your body converts any calories. It doesn't need to use it right away into the triglyceride. The triglycerides are stored in the fat cells. So, LDL cholesterol, the amount are these are the chart which represents. So, less than 100 is optimal, 100 to 129 is near optimal or above optimal, 130 to 159 is borderline high, 160 to 189 is high, and 190 greater than 190 is very high. Total cholesterol should be less than 200 for desirable, 200 to 239 is borderline high, and greater than 240 is high. HDL cholesterol, the less than low, 40 is the low, and this is your increase the risks. Okay. So, what are the drugs? So, statins are a class of the drugs that prescribed by doctors to help to lower the cholesterol level in the blood. By lowering the levels, they can help to prevent heart attacks, stroke. Studies show that certain people, statins reduce the risks of the heart attack, stroke, and even death from the heart disease by 25 to 30%. Statins, also known as HMG-CoA reductase inhibitor. So, when we were discussing this kind of cholesterol biosynthesis, polyketide biosynthesis, as I remember, I have mentioned there was a enzyme called HMB-CoA reductase, and statins are the inhibitors of this particular HMG-CoA reductase. If you see last week's PPT, you will find that there is a particular sequence for the cholesterol biosynthesis and the polyketide biosynthesis. There all the enzymes are mentioned. So, what are the example of the drugs? Lovastatin is sold under the brand name of Mevacor, which is among the others is the statin medication to treat the high blood cholesterol and reduce the risk of the cardiovascular disease. Lovastatin, a compound, is isolated from aspergillus and trias, which was the first statin to be marketed. We have simvastatin. It is sold under the brand name of Zocor among others and is a lipid lowering medication. It is used along with exercise, diet, weight loss to decrease the elevated lipid level. It is also used to decrease the risk of heart problems. In those heart risks, simvastatin was patented by Merck in 1980, came into the medical use in 1992. It is on the World Health Organization's list of the essential medication. So, statins or cholesterol lowering medications are also given for the obesity. Okay, these are also diseases basically are one kind of lifestyle disease. Okay, so these kinds of drugs are given for lowering the obesity. For the obese people, these drugs are given not only for the cholesterol, also obesity. Simvastatin is one of those. And then we have atorvastatin. It is a very important drug. Atorvastatin or Lipitor is a very important, one of the crucial drugs for the lipid lowering drug. This is included in the statin class of medication. By inhibiting the endogenous production of cholesterol in the liver, statins lower the abnormal cholesterol and lipid levels, ultimately reduce the risk of the cardiovascular disease. More specifically, statin medication competitively inhibit the enzyme a hydroxymethyl-glutaryl-CoA, which is the HMG-CoA reductase, which catalyzes the conversion of HMG-CoA to mevalonic acid. This conversion is a critical metabolism reaction involved in the production of several compounds involved in the lipid metabolism transport including cholesterol, low density lipoprotein, sometimes referred as a bad cholesterol, and very low density lipoprotein. So, this is the structure of atorvastatin. As you can see, we have the pyrrole ring, and this is 4 substituted pyrrole ring which has N substitution also. Then we have this rosuvastatin. This is also known as Crestor, used to with proper diet to lower the bad cholesterol and fats to raise the good cholesterol or HDL in the blood. And we have pitavastatin, which is usually a calcium salt, and it is a member of the blood cholesterol lowering medication class of statins. Like other statin, it is an inhibitor of HMG-CoA reductase, the enzyme that catalyzes the first step of cholesterol synthesis. It was patented in 1987 and approved for medical use in 2003. Okay, so this is the structure of the pitavastatin. So, one more thing I want to tell you right now is like since you are reading organic chemistry interface with the biological system, the thing I did not summarize was the type of heterocyclics which are present in each and every class of the drugs, because if you see, the heterocycles are very important. These are present as an active compound of the drug. So, remembering the names of the heterocyclic rings are also important. So, before examination also revise those things, like this is a pyrrole ring, this is a pyrimidine ring, this is a quinoline ring, okay, and these rings are the active cores of the each and every drugs. So, you can be given a particular drug structure and you can be asked to write what is the active core or what is the name present in the particular drug. So, you should be able to answer this kind of questions also. That is why you have to identify what is the particular class of heterocyclics present in the drugs, okay. If the structure is given, so you have to remember the names along with the structures. So, now antidepressants, we have a little bit about your myoinhibitors, right. So, this was a part of antidepressants. So, I told you I will just summarize this thing. So, what are antidepressants and how do they work? Antidepressants are a class of the drugs that reduce the symptoms of depressive disorders by correcting chemical imbalances of the neurotransmitters in the brain. Chemical imbalances may be responsible for changes in mood and behavior. Neurotransmitters are vital as they are communication link between the nerve cells in the brain. Neurotransmitters reside within vesicles found in the nerve cells, which are released by one nerve and taken by other nerve. Neurotransmitters are not taken by other nerves, are taken by some nerves and release them. The process is called the reuptake. Okay. The prevalent neurotransmitter in the brain specific to depressions are serotonin, dopamine, and norepinephrine, also known as noradrenaline. Okay, so this is the structure of serotonin. It is a very easy structure, a very simple structure. This is dopamine. Dopamine also has a very simple structure. Now, this is epinephrine or adrenaline, and this is norepinephrine, and which is noradrenaline. Okay. So, then in general, antidepressant work by inhibiting the reuptake of specific neurotransmitter, hence increasing the levels around the nerves within the brain, such as selective serotonin reuptake inhibitor, antidepressant that will affect the serotonin level in the brain. Monoamine neurotransmitters are the neurotransmitter and the neuromodulator that contain one amine group connected to the aromatic ring by 2 carbon chain, say like serotonin, dopamine, epinephrine and norepinephrine. These are the monoamine neurotransmitters. Okay, and so what are the classes of the antidepressant drugs? So, classification are the first one is the tricyclics or TCS and tetracyclics, like imipramine, doxepin, desipramine, amoxicillin. So, these are the examples. Monoamine oxidase inhibitor myoids like phenazine, serotonin, moclobemide, and these are the example of myoinhibitors. And then we have selective serotonin reuptake inhibitors, which are very important, SSRIs, which are fluoxetine, fluvoxamine, citalopram. These are the examples. So, these are the classes of the several antidepressants. So that is it. So, today this was our last session for this particular class, okay, and where we discussed different class of drugs, and we discussed about the questions which were in your previous classes. All these PPTs I will just share with you, and the today's class lecture video also I will share with you. So, I hope this course was beneficial for you. So, I will share my email ID even if you have any doubt after today's class. It is the last class for this course. So, let me just share my email ID. So this is our last class for this NPTEL course of organic chemistry in biology and drug development. We did about 12 week we did this kind of classes along with the question solving, and we discussed many things about the class, about this course. And today it was the last class. After that we won't have any this live sessions, but I will always be available through the email. If you have any query, any doubt regarding any course content, please drop an email to me. Okay, I will be happy to answer your queries, your doubts. And also your examination is also nearby, so I will hope all of you are reading the course materials which are given to you. So I hope you are you know where to find this kind of PPTs and the lecture materials. Just you have to go to the SWAYAM portal. Okay, just you have to go to the just I will show you where you have to go. So you are I think getting this online course of this like medicinal chemistry and biology and drug development. Just I will show you where is the your this kind of lecture PPTs and the videos are there in the SWAYAM portal. Okay, so what you have to do is you have to go to the online course of the SWAYAM portal. Just let me show you, so it will be easier to you. Okay. Yeah, I think here. So you are getting this kind of emails, I think. So you just go to the course section. Okay, you have to just I will show you where is the paper, this presentation, and where are the this kind of PPT and things. So you have to log in to the SWAYAM portal. Just let me show you. So this is the course page of the NPTEL Organic Chemistry in Biology and Drug Development. Here all the assignments are there each and every week. If you see each and every week the lecture, the feedback form and assignments are there. Here we will have this problem solving session. As you can see on the left side of the panel at the last one, you will have this problem solving session. If you click here, you will get the problem solving session recording, and if you click there, you will get this kind of a Google chart. Here all the links are present, the YouTube links are present of each and every class. Like for week 1 you will get the week 2, week 3, all the YouTube links are present, and in this YouTube links you will find our course lecture materials, and here the presentation folder, the Google documents are present here. If you click on one document, you will get like if I click here, you will get the Google slides which have the each and every course content, the questions which we did in the previous classes. So that's just this thing I wanted to show you, because many students are asking me so where to find this problem solving session and where to find the lecture material and videos. So just I show you, if you go to the SWAYAM portal, you will get on the left panel at the course outline, the left panel, you will get at the end the problem solving session recording, and you can find all the videos and lecture materials there. So thank you all for joining. It has been a wonderful journey with all of you in this particular class. The students, many students were actively participated in the class, so that's I am thankful about. So I think I will close the session for today, and if you have any doubt remaining, you can just drop an email to me. I will happily answer your query. Okay. So thanks a lot. I will close the session for today, and if you have any doubt, feel free to contact me, and all the best for your final examination. So thank you.